All right. Thank you, everybody, for coming to the third round of microTAStic virtual seminar series. It is so great to see so many of you joining us today. I'm Cindy Tang from Stanford, and on behalf of my co-organizers, Julia, Angela, and Bastian, we'd like to thank you for your ongoing support to make this seminar series possible. Just like the last round, we have an exciting lineup of speakers, and we have expanded from 6 seminars to 8 seminars in this series. This year, in addition to support by CBMS, we're very excited to partner with the journals Lab on a Chip and Microsystems and Nanoengineering, and continue a new format for a few of our seminars, including today's, where we'll have a short opening talk selected from outstanding papers published in the journals. One of the hardest things that the organizers have to decide is the time zone for the series. We understand that there's no time that will work for everybody. So to try to accommodate multiple time zones, in the first half of this series, which runs from today to February 2025, we'll have the seminar start at a time that is more amenable to Americas and Asia. While for the second half of the series running from March to June 2025, we'll shift the seminars to a time zone that is more amenable to Europe and Americas. We'll be posting more details as it gets closer. In addition, like the last rounds, we'll be recording the seminars if the speakers permit, and we invite you to subscribe to our YouTube channel for the recordings. Finally, I'd like to remind everyone of Zoom etiquette. Please mute yourself and type your questions in chat, and we'll do the Q&A at the end. Today, we're very lucky to have 2 speakers, Dr. Jungseub Lee to give a 10-minute opening talk based on his recent publication in Lab on a Chip, followed by Dr. Dave Weitz to give a 40-minute talk. So I'm going to unshare. Okay, and I'm going to invite Dr. Lee, if you're ready, please share your screen, and I will introduce you in the meantime. Dr. Jungseub Lee is a postdoc with Dr. Noo Li Jeon at Seoul National University. He specializes in developing microfluidic organ-on-a-chip devices using advanced 3D printing techniques. His work focuses on integrating image analysis algorithms and deep learning and computer vision to enhance analytical capabilities of these chips, supporting applications in drug screening and biomedical research. So without further ado, Dr. Lee, it's all yours.
Hi everyone, and thank you for the introduction. My name is Jungseub Lee from Seoul National University, and I'm honored to open today's seminar. In this presentation, I will be sharing an innovative approach to real-time analysis in organ chips, titled Integrating Deep Learning with 3D Cell Culture, Quantitative Angiogenesis Analysis using Virtual Staining Approach. To begin, let's first explore the foundational background of organ chips briefly. Traditional 2D in vitro models were limited in their ability to replicate complex environment. However, we now have advanced 3D models capable of mimicking intricate microenvironments, enabling high-throughput data generation in highly refined 3D microenvironments. While organ chip platforms have advanced significantly, analytic tools haven't kept pace. First, the large data output from organ chips is challenging to process quickly with traditional microscopy-based imaging systems. Second, most analyses still rely on endpoint assays, which restrict data collection to fixed point. This means that
Jungseub,
apart
can I
uh-huh.
stop you for a sec? So we see gray bars that show up on top of your slide. I don't know if those are might be Zoom. Okay, there you go. Yeah,
I
they're
think it
gone
is
now.
that,
Yeah,
yeah.
perfect. Yeah, thank you.
Okay. And second, most analysis uh, analyze still rely on endpoint assays, which restrict data collection to fixed point. This means that apart from a final endpoint, we are limited to using unquantifiable bright field images. Virtual staining technology offers an effective solution to these bottlenecks. So what exactly is virtual staining? Virtual staining is a deep learning technique that assigns color to label-free images, typically using a UNet architecture designed for image segmentation. At the bottom, we illustrate this process with a teapot example. The UNet encodes the teapot's patterns and textures, then decodes and reassembles these features to identify and highlight the teapot. This demonstrates the general approach in UNet-based virtual staining. Virtual staining has become a powerful tool without traditional staining procedure. Here we see examples of virtual staining applied to section tissue and cell images, producing results nearly indistinguishable from traditional staining. Let's examine how virtual staining impacts organ chip data analysis. In organ chip experiments, endpoint assay typically provide data only at the final time point, with bright field images for all other time points. However, these bright field images are often difficult to interpret and nearly impossible to quantify effectively. In contrast, virtual staining converts images into quantifiable data, allowing access to meaningful information. When we compare them by the graph, the endpoint assay data marked in blue only represent a single time point, whereas virtual staining marked in red allow us to gather data throughout the experiment. This enables high-throughput image processing and real-time quantification, making label-free virtual staining an invaluable tool for dynamic analysis. To achieve virtual staining, we need a large data set and a robust deep learning model for angiogenesis. For high-quality data acquisition, we use our lab's MV Impact platform, specifically designed for angiogenesis experiment. This platform provides a multi-layered hydrogel pattern that creates a 3D microenvironment. This slide shows the workflow for angiogenesis experiment. The cross-sectional illustration of MV impact demonstrates co-culturing cuvex and fibroblasts to induce angiogenesis. After 6 days of culture, we obtain 3D vascular structures. MV impact high reproducibility and rapid data acquisition capabilities enables us to collect numerous datasets from single chip. By utilizing multiple chips, the platform can provide enough training and test dataset for deep learning network. With this data, we train the virtual staining model using a conditional GAN architecture, comprising a generator and a discriminator. The generator, based on UNet, receives bright field images as input and generates virtual stained images. The discriminator then compares this with actual process images, evaluating similarity to optimize the model. Here's a simplified overview of the deep learning training process. First, the generator initially creates a virtual stain image from the bright field input, but early stage outputs are often unclear. And second, the model calculates the differences between the generated and real images using a loss function. Third, the difference is then fed back into the generator for parameter tuning. Then the UNet produces higher quality virtual stain images. With each cycle, the UNet moves closer to a fully trained model, and each loss function offers unique feedback to the neural network, affecting the resulting images. Let's examine the result of the virtual staining. I will focus on the 6 different loss conditions. Notably, the SIM plus GAN loss highlighted in red delivers the most accurate result. Let's take a closer look at the virtual stain imaging in detail. In the vascular network region, several images exhibiting blurring. And the vascular endpoint, many images suffer from blurring and deformation, making it difficult to clearly identify the endpoints of vessels. At the initiation site, some areas that should be open appear blocked. In the vascular mesh region, blurry and unintended gaps between vessels are common, potentially obscuring the overall structure. In the terms of the qualitative result, the SIM plus GAN loss delivers the clearest output, making it the optimal choice for maintaining structural fidelity. Now let's move to the process of quantitative analysis. We skeletonize virtual stain images to measure endpoint area and length, comparing these metrics across conditions. The SIM plus GAN condition closely resembles the ground truth, confirming its suitability for quantitative analysis. As an application of angiogenesis virtual staining, we compare vascular structure changes in drug treatment samples. The impact of sunitinib and bevacizumab was visualized and quantified, and we observed that the vascular changes in drug treatment treated samples closely match the trend in the actual ground truth data. Now I will show real-time virtual staining process. In this slide, I'm illustrating how we adapted a virtual staining model initially designed for high-resolution confocal images to work with real-time live imaging data. On the top, we see the original virtual staining model, which was trained on high-quality confocal images. These images allow us to accurately predict fluorescence signals without actual staining. However, this model was limited to endpoint images, meaning it couldn't be applied continuously over time. The key step here is model adaptation. By tuning the model, we enable it to process live live bright field images from high-speed microscopes. This adaptation allows us to produce virtual stained images in real time. Here, we can illustrate our final result through virtual staining in real time. First, we capture bright field images of live samples inside an incubator, generating a time-lapse video. By applying virtual staining to these images, we transformed them into fluorescence-like quantifiable images. This allows us to analyze the data continuously, unlike traditional methods that only provide snapshots at single time points. As shown in the graph, we can now track dynamic changes such as vessel growth in real time. This approach gives us a complete view of the biologic process. In conclusion, this study aims to establish a real-time label-free virtual staining system for angiogenesis, generating virtually stained data from live cell bright field imaging. By combining a high-throughput organ-chip platform, a deep learning-based virtual staining network, and real-time quantification algorithm, we enable dynamic, continuously visualization of 3D vascular network. This system enhances continuously label-free quantification of dynamic processes, addressing a bottleneck of traditional analytic method. And I'd like to thank my advisor, Professor Noo Li Jeon, for his guidance, and my lab members at MBEL for their support and collaboration. Thank you.
Great. Thank you so much, Dr. Lee. That's a wonderful talk. So I encourage everybody who have questions to type your questions in chat. I think for the sake of timing, we'll do all the Q&A at the very end after both speakers' talks. So Dr. Lee, if you don't mind unsharing your screen, and then we can have Dr. Dave Weitz share his screen. And in the meantime, I will start introducing Dr. Weitz. So our second speaker is Dr. Dave Weitz. He received his PhD in physics from Harvard University and then joined Axon Research and Engineering Company, where he worked for nearly 18 years. He then became a professor of physics at the University of Pennsylvania and moved to Harvard at the end of the last millennium as a professor of physics and applied physics. He leads a group studying soft matter science with a focus on material science, biophysics, and microfluidics. Several startup companies have come from his lab to commercialize research concepts. All right, without further ado, Dave, it's all yours.
Okay, well, thanks very much, Cindy, and it's a pleasure to address you all. I haven't given a virtual talk since COVID, so we're back to that. But anyways, it's really a pleasure. So I want to tell you some work that we've carried out over the years in my group. Some of it's new, quite a bit of it's new, actually. A lot of it's quite a bit older. But it sets the stage for everything. And we're big fans of droplet-based microfluidics, and I want to describe some things that we're doing, mainly for biotech applications, but it's trying to understand some physics as well. And the work I'm going to tell you about is primarily due to a current student in the group, Xing Zhao, and a postdoc in the group, Kevin Yanke, but we have contributions from the people I've listed, plus many others as well. So let me start with some sort of biotech applications and just tell you something to put things in perspective. We are now doing a Zoom call. And the last time I did Zoom calls was during COVID when we had to teach that way. And so I ask you to go back and remember the horrible years, the early years of COVID. And we all, at least at Harvard, we had to get tests a couple times a week to see whether we had COVID. And the way you do that is with a PCR. And so PCR is the gold standard of detecting something very sensitively, detecting very low levels of basically genomic materials. And it's a wonderful test. It can be done relatively inexpensively. It's very fast. It's very, very sensitive, very, very specific. But it's limited in the sense that you can only detect one, or maybe two, or maybe a few different samples at the same time. You're using some fluorescence detection, and you don't have that many colors. So you typically only detect one thing at a time. And I want to think more broadly about PCR and think about how can we expand the way we can detect things. So the first thing that I'm going to do is I'm going to focus on what I call digital PCR, digital droplet-based PCR. The concept is very simple. You take the sample, you compartmentalize, we compartmentalize it into drops, and you do the PCR reaction in each and every drop. And if there's a target in the drop, you get a positive response. So that's why it's digital. It's either 1 or 0. And you just dilute things enough so that you only have 1 or 0 target oligos per drop. Then it's much easier. You don't have to do any calibration. You want to know how much there is, you just count the number of positive drops. The sensitivity is basically limited by the background fluorescence. The dynamic range is set by the number of drops that you use. And so typical numbers that we use, say 25 microliters of sample, that's a typical volume for a PCR sample. We might make 1 million drops. Yeah, let's say your sensitivity is 10 drops. It should be 1, but let's say it's 10. And then your 10 molecules per 25 microliters, I get that to be attomolar sensitivity. Very, very sensitive. But as I say, you can do it with digital sensitivity. You can target single oligos. I want to ask, can we somehow multiplex it? Can we look beyond what you normally do? Now you can do this by, you just use the fluorescence, use a couple of colors, you can get a couple of samples. Can we do something else? And so I want to introduce what I call double drop digital PCR. This is really something that Huidan Zhang, a former postdoc in the group, really conceptually introduced quite a while ago. And the idea is you do digital PCR, you use fluorescence to detect whether it's a positive drop, but you don't necessarily identify what's in the target. You do a different experiment then to identify what the target is. And I'll show you some simple ways of doing it, and then I'll tell you some more sophisticated ways that we're trying to work on now. The advantage of this, you'll have improved, not only will you have multiplexing, but you have improved sensitivity, and you can really identify things very clearly. So here's the first example. This is really what Huidan was trying to do when she first conceived of the idea. I mean, I think we now generalize it conceptually beyond what she was doing, but let me tell you what she was doing. She was trying to detect mutations, KRAS mutations, and the trouble is there are 12 different mutations in one codon. There's a bunch of different mutations. You'd like to really understand whether they exist, and how frequent they are if you're trying to understand something about the progression of various diseases. And the most important one is metastatic colorectal cancer. So what Huidan did was she looked where the mutations were, and she took a peptide nucleic acid, something that will bind to all regions in the wild type. So it'll bind, because it's a nucleic acid, you can get it to bind to all the regions very, very firmly, and that will prevent PCR. It's clamped there, and it prevents PCR. So if you have a wild type, you can do the amplification, but you won't amplify PCR, you won't amplify the wild type. And this is, of course, immediately valuable because you're looking for a very small number of mutations in among the wild type targets. So the first thing you do is just measure the mutation load, and you just do it with and without this clamp. Without the clamp, then you amplify everything, and you measure all the KRAS that's there, and there's really a small fraction that is the mutated. But then you put the clamp on, and you do the experiment again, and you measure only the samples where there are mutations, far fewer. And just by counting, you can measure the ratio of mutated to total, and you can measure the concentration of both. You measure it directly since you're doing this digital PCR. And so this way you can detect what the mutation rate is, but you don't detect what the mutations are. But the point is that those drops that have been amplified, they have now many copies in a very pure amplification, many copies of the mutant KRAS. So you can sort the drops and collect only those positive drops. And then you can combine the emulsion together, and now you have a very cleanly amplified version of all the mutations, because you've amplified all the mutations. And you have millions of copies of that. So now you have plenty of material to just divide this up and make a separate digital PCR for each of the mutations, where you use primers that are specific to the particular mutations. And so when Huidan did this, it was very easy for her to identify, for example, this particular sample had these two specific mutations. So she's measured a mutational load, and she's measured exactly what they are. And conceptually, the way it's done, the way she did this, was to separate the readout of the positive drops from exactly what they are. That's why I say it's double digital PCR, because you do it twice, but you do the second round on the amplified version of the first round. You can do this now, or you can use this concept to do many other things. For example, if you're looking at something at a very, very low mutation rate, or a very, very low population rate, you then want to really do something that will get rid of all the noise. If you have some amplified signal from some noise or something like that, you can do the same thing, but now you can use nested primers, so that the first set of primers amplify something over a somewhat longer range of the amplicon, and then the second set will just take somewhere on the in between. So if you have noise, the second set won't measure it again. So this is a way of validating, of reducing the noise in the signal, and you can get better signal, better selectivity, and better sensitivity. And so here's some experiments that Huidan and Naiwen Chu did, where they tried to detect very low concentrations of bacteria, and did this by just looking, in this case, at the 16 RNA of the bacteria. And this was just spike-in experiments, and it's a little difficult to see, but if you look carefully, you see that they're spiked in down to a single bacterium in something like a mil of fluid. And this is sort of the concentration that you need to really detect very, very low concentrations of bacteria if you want to do something like looking for sepsis. And you're really able to get good signals down to very, very low concentrations. And so we worked with Deb Hung, who just went to the hospital, and she got some samples. This is of staph, And she got some samples. She said, give us 4 samples. 2 are negative and 2 positive. Please, can you tell which are negative and which are positive? So we did the 2 negatives. We found they're negative. But of the 2 positives, we only found 1 was positive and the other was negative. That's what we said. Well, Deb went back to the hospital, looked more carefully, and in fact, only 1 of them was positive. So this works on real samples as well. But now, this is a very nice thing, but you have to do a whole second round. We asked, could we somehow make it more generic? Could we find some way to do a second measurement to read exactly what's in each of the drops? And what we have devised, and this is really the work of Xing Zhao, is a method to use melting temperatures as a way of barcoding both the primers and the target. So let me give you an example of what this means. So the melting temperature is nothing more than if I have a piece of DNA, it's double-stranded. If I heat it up, eventually it melts into 2 single-stranded regions. This is exactly one of the steps in PCR. You have to take double-stranded DNA and make it a single-stranded DNA so it can be multiplied. But the profile of the melting temperature is really well defined, and the exact temperature that it melts at depends very sensitively on the type of bases, because each base pair has a slightly different energy, and the way they're ordered, the way they put together is it determines the melting behavior. And so here's, for example, what you do is you just use a dye like evergreen or something like that, which will go in and it fluoresces when it's double-stranded. It doesn't fluoresce when it's single-stranded, and you just measure the decrease in the fluorescence. It might look like something like this, and you want to find exactly where the melting temperature is. And if you take the derivative, you get a peak in the curve. And this is something that's actually well established, and people do a lot of measurements just using the melting temperature, and you can actually determine, even in some cases, you can determine the sequence of that just by knowing how these things melt. So what we do, or what Xing does, is now tries to develop for the primers, she tries to develop a series of barcodes that sets the melting temperature. So we have the melting temperature of the target, but we have the barcodes as well. So we can on one side, we can put a barcode that has a lower melting temperature. On the other side, we can put a barcode that has a higher melting temperature. And so, for example, if it's a higher melting temperature, we might have the target with some peak, but we can have a second region that has melts at a much higher temperature, and we can adjust what that temperature is just by adjusting the sequence in the primer. And so this gives us a way of getting additional barcodes that are built into the primers. So we can have primers that are designed to target specific molecules, and we barcode them by their melting temperature. Well, this is not a bad way of doing it, but it turns out that if you calculate how many melting temperatures you can distinguish, it's not bad. You can probably get 10 or 15 on one side, the same on the other side. So you have now, a number of barcodes. If you can do 10 and 10, you have already 100 barcodes. But really, we wanted to do even more than that. So in addition, what we did was we also, when we do the amplification, we do an asymmetric amplification. We use more of one primer than the other. So sometimes you get double-stranded, but sometimes you get single-stranded DNA as well. And in those regions where we've defined the melting temperature of the primers, we also use a set of probe molecules. These probe molecules will bind to a certain region at each of those high and low melting temperature. They're a fret pair, so when they're bound, they fluoresce, but at some temperature, which we can adjust very precisely, they unbind and the fluorescence decreases. So now we have a completely orthogonal, a completely different way of using a barcode. And because we're using fluorescence, we can use a different color. So it can be completely orthogonal to this. And now we have a whole new set of barcodes. And so now when we do this, we have the two temperature barcodes that are of the primers themselves. These will come from the double-stranded DNA, and we have the series of barcodes, the colored barcodes, that come from the probe molecules. And we have all of these things, and we have the product of all of those things. And this is a fairly old slide, but if you just calculate all the different numbers, it's something of order 3,000. I think now Xing is pretty convinced that she can make something of the order of 500,000 different barcodes by very precisely controlling these probe molecules and adjusting the temperature. And these are really completely independent of everything else. So you have tremendous control over them, you don't need primers. It's a very simple way of doing this. So now you have a way of barcoding a huge number of samples just by doing the experiment. So you put them in one drop, you do the amplification, and then you do a melting temperature profile at the end. I'll just show you some examples of how it works. These are actually not done in drops, they're done in wells, just because we didn't want to deal with getting everything to be completely thermally stable. Ultimately, we'll go to a drop, but we wanted to do a proof of principle. And here, these are the different barcodes. This is the one temperature barcode here, and these are the second set of barcodes where we've adjusted the temperature. You can see the peaks are really well defined, and you can clearly measure them and distinguish them. And we're only trying to measure them, say, a degree apart. We probably can do half a degree apart. And so here's a case where now we include not just the two end barcodes, but we also include the two color barcodes, but these use the same color, the high and low temperature. And this is a case where we can identify quite easily something like, I think there's 64 different barcodes, and more recent data, Xing has done even more than that. You can do this really very precisely, and you can identify many different things. So now this really is something that sort of takes you right between two ways of doing detection. So normally, as I said, if you want to do high sensitivity, super high sensitivity detection with really the gold standard in sensitivity, you do PCR. It's fast, it's cheap, and it's very sensitive. But if you want to detect many, many samples, what you typically do now is just next generation sequencing. Next generation sequencing, you get a huge amount of data, and you just start counting the particular sequences that you're interested in. And because you have so much data, this is actually a powerful way of probing things. And now there are many diagnostic tests, and more importantly, at least to me, there are starting to become many public health screening where you look for large number of markers, large number of mutant markers to look for propensity of disease, to screen in advance. But this is something that gives you huge amount of multiplexing. But sequencing, even as it gets cheaper, is still not that cheap. You still need to do many rounds of PCR just to prepare the sample. The sample preparation is much more complicated than a PCR experiment, and it's really going to be much longer and higher cost. So this is good for doing screening for a wealthy company, a wealthy country where you have time, but it's not really going to be the best way of doing public health screening. If you have something now that combines both of these things, then I really see this as a potential way of using what you do with next generation sequencing to screen many, many samples, but doing it with the same sensitivity and specificity of PCR, but with the multiplicity, the multiplexing power of NGS. So there are many other ways that things that you can do. And again, conceptually, I would just say, look, what we're doing is we're doing something to detect. Usually, in this case, what I've showed you is PCR to detect, something to amplify, that's PCR, and then something to detect what you've measured, to measure, and that's the melting temperature. But you can do the same thing. If you can use DNA as some way part of the detection. The amplification can be PCR, and then the final detection can be this melting temperature. So another possibility is just to use to do a single protein detection, and for that you can use a pair of antibodies and have pieces of DNA on each of the antibodies, and you ligate and amplify, and then you just this amplified region of the DNA just becomes your amplification scheme with PCR, and the readout becomes the same kind of melting temperature barcodes. And so you can do now single molecule protein detection, but that's something that's multiplexed. And so this idea of detecting this way is done commercially, but then the readout is all done by sequencing. And the idea of detecting single molecule of protein is also done commercially, but the readout is something like an ELISA, which doesn't have the ability to multiplex. So you have the ability to do some combination of very, very sensitive detection with high multiplexing. This is just an example where Xing did IL-10, just a standard protein, and she showed that she's sensitive enough that she's well below, she can detect the concentrations well below physiological limits. And actually, it's sort of interesting because now you have additional ways of getting rid of the noise. You're not just reading the barcodes, but you know the melting temperature of the combined DNA as well. That's an additional piece of information to make sure that you're not having some random combination of two DNAs and you're getting some amplification of something that's noise. And that the same thing happens when you're detecting molecules. You can use the melting temperature of the molecule itself as a validation to reduce the noise. So this is a very, very highly sensitive method in all respects. Okay, let me say that this was my application of purely biotech. Now I'd like to still talk about biotech applications, but really look a little bit more in the realm of trying to understand some biophysical things, trying to make something with droplet microfluidics. Here, we're using the droplet microfluidics to do reactions and take advantage of the multiple reactions that we can make. Now I'd like to go back to the droplets themselves and ask, what can we do with the drops? How can we use them in some way that we take advantage of the drops? And again, that's something that we've worked on over the years because you can make drops of one fluid in a second so precisely, you can use the drop structure itself as a structure, the template to build other structures. So again, we're thinking about something that's also, as was this last experiment, as I motivated it from the terrible days of the pandemic. This was also motivated by the terrible days of the pandemic, and thinking, you know, when we live in Boston, it was amazing during the pandemic. It was really amazing that within a year there was a vaccine for COVID that was developed and available. I remember getting in line, just logging in late at night to try and get the vaccine. It was developed in Boston by Moderna, and it's basically these mRNA vaccines. But a key to the mRNA vaccine, it's not only to get the mRNA correctly to work, not only to get it to stable, but also to encapsulate it. And people use liquid nanoparticles for encapsulation. And this, again, is a wonderful, wonderful technology that is very, very effective for mRNA. But we asked some questions, what are some limitations? How can you go beyond this? And because of the success of the mRNA vaccines, there was a renewed interest, certainly around here, in trying to understand what kind of drug delivery vehicles you can do for these modern therapeutics. So the first thing that we did was we designed a way of mixing two fluids together to make LNPs, and I won't show the details of it. It's basically a turbulent mixer. It turns out that you can buy even commercially available microfluidic manufacturing systems for liquid nanoparticles. They're not going to be something that you can do billions and billions of vaccines, but they are something you can do quickly. You can formulate, you can test, and for doing something where you don't need to make a huge amount of vaccine, they're still efficient. They use microfluidic mixers that were designed like 20 or 25 years ago by my colleagues Howard Stone at the time and George Whitesides. But really, we should be able to do better. And so we designed our own turbulent mixer, a microfluidic turbulent mixer, and we were able to get very, very fine particles. We could make hydrodynamic diameters down to really, we could control it down to easily 40 nanometers, up to 160 nanometers, just by controlling the flow rate, and the polydispersity of these things were very good. So we were just trying to convince ourselves that microfluidics would be applicable to a system like the lipid nanoparticles. But really, if you think about LNPs, they're fantastic. They're really exceptionally effective. But the way they work is you take the phospholipids out of some solvent, you crash them out of some solvent, and you put the drug in the solvent as well. So it has to be a drug that will withstand the solvent and that is bound by charge. It's coulombically interacting with the LNPs to load the LNPs. And this is great, but what if you want to do molecules that aren't charged like that? Well, there are many other delivery vehicles. One of the most common and widely investigated is just using viruses. They're extremely good delivery systems because that's what viruses do. They deliver something into the cell. The problem is, first of all, you can get immunogenicity. The body can become, the immune system can start to identify it, so you can't use it over and over and over again. Also, the size of viruses are relatively small, so you can't really put everything you want inside a virus. And so we were asking, could we deliver larger payloads? And could we deliver them without the charge requirement? In fact, I have friends who, they went to work for these modern protein delivery companies, such as CRISPR-based companies, where their whole goal was just to shrink the CRISPR so it would fit in a virus. And it just seemed like the wrong thing to do, to change the therapeutic to fit into the delivery package rather than to adjust the delivery package to match the therapeutic. So we tried to do something different. And so we took a very old method that we developed in the lab many, many years ago, and we tried to put a new twist on that, taking advantage of all the things that we've learned with microfluidics. So the old method was something that Sophie Pateau developed with Philippe Poulin and Barbara Friskin. This was when Sophie was a student a long time ago in the group. Barbara was a visitor. And they really had a very simple and clever way of doing things. They made an emulsion, which is a water and oil emulsion, where they put a phospholipid on the outside of the surfactant, and then they passed it through a second phase of oil where there was an interface with an aqueous phase, and there was a second type of phospholipid. It could be the same as the first phospholipid, but it didn't have to be. You could use something that was different. And so when it passed through, you could get something that was an asymmetric structure. And Sophie worked very nicely, very beautiful technique. The problem was it didn't really work so well because she was doing it with fairly large vesicles at the time. They were something of the order of 10 microns or so. When it worked really well, but it really wasn't a very effective way of doing things. In fact, this is one of the reasons that we were inspired ultimately to try and make things with microfluidics, because the early days of this was before we were making drops with microfluidics. So now Kevin Jahnke takes the same idea, but he just does everything on a slightly smaller scale. So the first thing he does is he makes this emulsion where the phospholipid is the surfactant, but rather than just making a large-scale emulsion, and rather, in fact, than using, say, a single microfluidic channel, which you're sort of limited in terms of the size, the small scale that you can get to just from clogging reasons. Instead, he uses a membrane. He uses the same thing that you would use to extrude vesicles. It's basically a membrane with very, very controlled size. You can buy these, they're made with damaged paths with neutrons, and so they're very, very well-defined, and he extrudes the emulsion through this. So this is really like nothing more than a membrane emulsification. There are many, many holes, a very fine controlled region. You can make an emulsion, and you get these large emulsions. This is before extrusion. You extrude them. You get really a very well-defined, this is measured with light scattering. You can get a very well-defined small scale. You can control the scale by controlling the size of the membrane. This is something that certainly is scalable because it's really just the same as membrane emulsification. And then he takes these and he uses the same method, the same conceptual method that Sophie developed, and passes them through a second membrane. Now, of course, these are much smaller, so you can't just use gravitational forces, you have to centrifuge. But what's interesting is because they're so small, at least this is my belief, they're so small, the curvature here is much smaller. It's a much more efficient process, and he gets much more efficient production of these vesicle-like structures. And here's an example where he's used now phospholipids of all different types. He used charged and uncharged. He pairs them with the inner and the outer leaflet, and he doesn't get perfect efficiency, but the efficiency is not bad. And now we can start asking sort of what are some of the biophysical reasons why are they more efficient. And so what he did was he measured the surface tension, and he showed that if the surface tension between the interface here is lower, then you get more efficient production of the structures. But still, if you're trying to make something, you can afford, if you're making something valuable enough, you can afford to have not 100% efficiency if you can make some interesting structures. And here's a way now of really making asymmetric vesicle structures, asymmetric liposome structures. You can test that whether they're asymmetric. You do that by putting something that will bind a dye on the outside or the inside of the vesicle. You bind the dye and then you quench it, and you just see does the dye bind on the outside and you see fluorescence, or does it bind on the or is the bound region on the inside and you don't see fluorescence. And he shows that you can get very high degrees of asymmetry using this method. And you look at these asymmetric structures, and they are taken up in the cell. These are HEK cells. And if you look at these things, I'm sorry I don't have this repeating, but you can see that they're moving around inside the cell. So they've been uptaken, they've been moved into the cell. And you can also measure this, and what you find is that if you use asymmetric structures, they're uptaken much more effectively by the cell. And we're not exactly sure about why this is, but we think it's some combination maybe of changing the stiffness, but maybe matching better the asymmetry of the membrane of the cell itself. But you now have much more control because you can use, you can change the composition of the structure on the outside, on the inside. You can have something that will, say, interact coulombically with the cell if you want to, or you can have something on the inside that's different. You have more flexibility in terms of what you can do. And if you put something that actually gets released, so you ask, well, can you not only get the liposome taken up in the cell? You saw the fact that they're moving. We think that it's very obvious. We think that the uptake is through endocytosis. So the cell forms a little membrane, and that membrane surrounds this liposome. So you have, in some sense, a 3-layer membrane. But now you have control over that. What you want to do is somehow release the cargo. So what Kevin did was he used some mRNA that transfects and produces GFP, And he just shows that, in fact, you can you can transfect the cell and you can get GFP. And by looking at the different combinations, he can find that combination of, in this case, different charges, the inside and the outside. You can find the combination that both is uptaken in the cell and transfects the cell with the highest efficiency. Also, what's interesting is that the cytotoxicity, the cell death due to the presence of these liposomes, they always are somewhat harmful to the cell, but the cell death is greatly reduced using asymmetric structures. Then another thing that Kevin did, and this is just sort of a beginning, he said, well, look, normally you coat liposomes with something like PEG to make them so-called stealth, to get them, to prevent them from being taken up by the immune system. But what about coating them with something else? What about, in fact, coating them with a polysaccharide? And the reason for doing this, the rationale that Kevin had was polysaccharides might match the glycocalyx of the cell, and you might be able to target different cells by putting different polysaccharides on the surface. And so he basically devised a scheme where he binds the polysaccharide to cholesterol, and the cholesterol then he adds this after he makes them, or while he's making them, and the cholesterol binds into the membrane, and the polysaccharide is on the outside. And he shows that this is also a very effective way to get these liposomes into the cell and to transfect it. This is just looking in the cell, and by both tuning by using fluorescence for the liposome itself, you can tell that the liposome is uptaken by the cell. By using by transfecting for GFP, you can see whether the cargo is released, and then by coating it with this polysaccharide, you can measure the effectiveness of how it works. And what he finds is, in fact, that the polysaccharide coated liposomes work much better. Their transfection efficiency is much higher than when they're not coated. And similarly, the asymmetric structures work much better. So this is a way of combining both a new way to make a structure that's better, that's lower cytotoxicity and better uptake, and whose size you can control so you can encapsulate. You're not limited in what you can encapsulate, and where there's some potential for targeting just by having a better interaction of the coating with the cell. And this is just looking at different amounts of coating with the polysaccharide, showing that the uptake increases and that you can sort of target different types of cells by using different coating methods. Okay, with that, let me stop. I've tried to show you some combination of what we can do both towards biotech and towards materials, where there's a lot of interesting biophysics because of the materials as well. And it's all taking advantage of the power of being able to use microfluidic techniques to make for us to make droplets. And so let me thank you for your attention, and let Cindy take over with questions and answers. Should I stop sharing my screen, Cindy, or?
Sure, yeah. Thank you so much, Dave. This is such a fascinating talk. It's always so exciting to see all the wide range of projects in your lab and how they have moved so far into translational research. So we have a lot of questions in the chat, and I see that Jungseub has been answering them really nicely in the chat. So I will maybe see which one hasn't been answered. It seems like all the questions for Jungseub has been answered, so maybe I'll start with the ones for Dave. Navid, are you still here? If you're here, you can unmute and ask your questions. If not, I will ask for you. All right, so I don't see.
Hello, can you hear me?
Yes. Yes, go ahead.
Hello, Professor Weitz. Thanks for your great presentation. It was an honor to participate in this presentation. About the section of, digital ddPCR. Chance for next generation sequencing on this platform. Also for point of care, do you think this platform can be used for point of care? And what are the limitations? Both for point of care, you said that it's a really cheap platform. So is there any chance to be used on point of care? And also you said it has sensitivity like NGS. Is there any chance to do the NGS on it or something like NGS?
Okay, so those are good questions, Naveed. So first of all, in terms of NGS,
Thank
it is not a discovery platform. NGS will sequence things where you don't know what they are, and this will not replace that. But it, as a detection platform, I think it will compete very well with NGS. And so my goal, my dream, is to put NGS out of business for detection, because I think this is a much better way to detect things that what you would normally detect with sequencing. Of course, I have to dream big, which I do. I'm not convinced that we'll do it, but we'll certainly try. As to point of care, yes, you could do this in a point of care situation. I would say that it's not the ideal system because you have to do PCR, so you have to change the temperature. Maybe you could do with isothermal, but it's harder to design primers. But it's very often with point of care, you're not necessarily interested in really high multiplexing. So you might be able to, with point of care, if you could do this all with a low multiplexing and maybe just a couple of different detection systems for real point of care out in the field, that might be a better way of doing it. And I know people who are trying to design PCR-based systems that do that. If you wanted to make this point of care for clinics or something like that, absolutely, there should be no problem. You can make a very small device to do the readout, and the actual sample preparation is not so difficult.
I saw that sensitivity was around picogram per milliliter. So I think the troponin also is has the same sensitivity. But you said it can be for attomolar. However, the presentation shows the sensitivity for the picogram, I guess. Is there any certain reason that the sensitivity was limited to picogram?
I think so. I'm not adept at converting from molecular weight to concentration. I believe my concentration calculations, I think you could probably do better than that for concentration, but not a whole lot better. And that's just a counting argument. For the detection of the molecular weight, I'd have to convert. And I'm sorry, I just don't normally think that way.
I mean the concentration of the. I think that you showed us the interleukin-10. The concentration was
okay.
around.
So, but you know that's. There is some amplification or concentration of the signal, which you would normally do in something like that. And so I think we were just trying to do a proof of principle, a proof of concept. I don't think we are trying to make it as sensitive as you could. And I think there are probably ways of really improving the sensitivity, but that's the front end before the detection.
Yes. What's the time? That's
so sorry,
my last
Navi,
question. I'm sorry. Yes, I'm
quite
sorry.
a lot of questions.
That's my last question.
Maybe
I'm sorry. Yes.
so
Yes,
we'll
I'm
move
sorry.
on to
The
the
last
next
question, what is
one.
the timing for the
No, I think
for
David,
the
let's move
PCR,
on.
you know?
No, let's move
Yeah,
on.
let's move on because
Yes.
we do have a lot. And if you have time, maybe we
Yes,
can do
Thank
after
you,
the
thank you.
hour. Thank you. So I have a
Yes, thank
question
you. Thank you.
from
Sorry, sir.
Mushtaq. If Mushtaq, if you're here, you can unmute. If not, I can read your question.
Yes,
Wait.
can you hear me?
Yes.
Yeah, thank you so much for Professor for your insightful talk. First of all, I'm assuming that you have used droplet passive droplet protection such as T-junction or flow focusing device for droplet protection and encapsulating the cells like the DNA or other things for the like PCR. So these devices often yields numerous empty droplet or droplet containing only barcoded beads or droplet containing only cells or lot of cells within one droplet. So these are the few limitations mentioned in the literature. So I am curious to know that whether these problems, such as many empty droplets, many droplet barcoded only beads, many cell only droplets, so whether these are the problems in the real applications such as PCR or not. So can you please elaborate?
Yeah, I mean, I think you're pointing out some very important problems. They aren't problems in the samples that we're using for the PCR because we're not doing cells and we're not doing beads. If they were, we would certainly have to do something to avoid that. And you know, again, that's a totally different talk. I don't think that we have any fewer problems than everybody else. I guess we're somewhat partial to flow focusing when we make devices rather than T-junction, and we've developed different ways of trying to improve the encapsulation of cells. But you know, if you want to have one cell in a drop, you have to go down to very low concentrations. That's the only way that we know how to do it. There's ways of if you want to have a bead in a drop, there's ways of having higher concentration of single bead drops. That's something that Adam Abate developed, and there's ways of doing that. So all of these are practical problems. I don't have any foolproof way of solving them, except that I'll say this, that my students who actually do the experiments get fairly adept at avoiding them. But if they can avoid microfluidics at all completely for drop making, they do. And so again, Adam showed that you can, if you use beads, you can just shake an emulsion and make uniform droplets. And people have tried to copy what he does there a lot in our lab. So these are just practical experience.
Yeah, thank you, Professor.
Right, so I will note that we are 1 minute past the hour, so I just want to officially end this first seminar and thank both speakers for a wonderful opening talk. And I would, there's still a lot of questions in chat, and if Jungseub and Dave, if you have maybe 10 more minutes or so to stay for more Q&A, that would be really wonderful. All right, thank you everybody, and I hope you can join us in the next seminar in a month's time. Thank you.